So, good afternoon. It's already afternoon. I have a flu, and I apologize for speaking like this. Yesterday night at the dinner, I couldn't speak at all, but today I'm doing better. And thank you very much for inviting me and letting me share with you some of the research that we do in our lab. And as Ronnie said, part of my group is interested in cancer. And in order to explain what we were looking after, I would like to give a short introduction about DNA. Many people probably know about it, but I thought that it will be good to review it so you will understand what's going on in cancer. So what we see here is the double helix of the DNA, and it's a language. It's like any other language, which is built by letters. The English language has 26 letters. The genetic language has only 4. Very simple. You may know this: A, T, G, and C. These are the letters. And as in languages, that the first information is in words, in the genetic, it's the same. In our languages, the words can be in different lengths, from 2 letters to whatever in German can be 20 or so. In the genetic language, it's always 3. So we have 4 letters, and each word is composed of 3 letters. And as in languages, the message, the first message, is within a sentence. If we want to deliver a message, we say a sentence. And in genetics, the combination of the words that is the sentence is called a gene. A gene for eye color. We are all convinced that eye color is a genetic trait which is encoded in the DNA, is a sentence which reads what will be the eye color. So here is the gene that we just described, which is a sentence written in our DNA. And the DNA, the double helix, we do have 23 from our mother and 23 from our father. So we have 23 pairs, and they are packed into a structure which is called chromosome. And I'm sure that you have heard about our chromosomes. So here is a way that we can look in the microscope on our chromosomes, and I am very proud, although it's not my work at all, that the development of this technology was started in Israel, but by very talented physicists. And they have established a technology and a company, and they developed a way that we can see, or we can stain each of our chromosome in another color. So we can see here chromosome number 1, the 2 that we got from father and mother in yellow, and number 2, etc. And you can see how we see them under the microscope before we put them in pairs. But this process of the genetic material can be disrupted. And the disruption can occur when the DNA is duplicated. We all know that we, as human beings, all are starting from one cell, right? An egg and a sperm fused, and we have one cell. And the cell is dividing again and again, and then we have the baby. Okay, so in this, when the cells are proliferating, because we need the DNA in each of the cells, the DNA has to be duplicated or replicated. That's the word that we are using. And this process may have mistakes. And as you will see, this is the basis of cancer. So under normal conditions, sometimes there may be mistakes, but they will be corrected. So let's see what kind of mistakes we are talking about. So it can be a change that it should have been A, but when it's duplicated, it became a G. Think about the language. If a word will change one letter, then it may lose the message, the sense. It can be an insertion of another letter, a deletion of another letter, etc. And another very important process that may occur and does occur in cancer development is breaks in the DNA. If there is a break, it doesn't work so good. Okay, if there is a break here and a break here in the DNA, so in many occasions it will be joined correctly. But it may be inverted. So the small piece that we can see here may be inverted. And if there is a gene here and a gene here, the information will be cut and disrupted. Another consequence of breaks that we do have normally on very, very low level, but it's a major process in cancer, is that we will have a break in one chromosome, a long one in this example, and another break in another chromosome, and then they will fuse or join incorrectly. So let's see what happens in cancer. Cancer has, unfortunately, many, many features, and they are mentioned here. Recently, we became aware that there are more features of cancer, and one of them is that the DNA is unstable and there are changes. So we do have instability, breaks, and all the consequences of breaks that I have shown as examples, and also changes in the letters, which we call mutations. So these two are hallmarks of cancer. They occur very early in the process, and they promote cancer development. So we can see here, we have seen before a normal cell, and each chromosome has one color. Unfortunately, in a cancer cell, you can see what we talked about. We can see this chromosome, which has light blue and dark blue instead of being either this or that. And you can see that there are many chromosomes here that were rearranged. We call it's unstable. There is instability. Okay. So the question that we in the lab asked is, what is the basis, what is the molecular basis for this instability in early stages of cancer development? And this project, initiated by a very, very talented PhD student, Asaf Bester, who is now in Boston for his postdoctoral fellowship. And actually, Asaf suggested that we will try to investigate this question in a specific cancer. And you will see that it applies to most cancer, but we started with this, which is cervical cancer, one of the most common cancer among women. And we know that the cancer initiates by the infection. So Ronnie said that we will not be talking about infection, but actually we do, because the infection is changing the genetics here. We all know that cervical cancer starts by the infection of a virus, human papilloma virus, either 16 or 18. And the reason that we decided to start and ask the question in this system is because we know the starting point of the process, which is the infection, and we know which genetic genes, genes that the virus has, are initiating the process. Okay. So we had an hypothesis which was counterintuitive, turned out to be correct, but was very counterintuitive, and it is based on what we know in our lab for many other studies that we did. We hypothesized that we get breaks in the DNA because something is wrong with the DNA duplication. It's the DNA replication, that there is stress, we call it. Something is not optimal. So think about it. A cell is driven to proliferate in an unplanned way. It's abnormal. So on one way, the cells are now proliferating. They need the DNA to be duplicated, and our hypothesis is that the breaks and the instability is because the DNA duplication is incorrect. So I would like to share with you the technology, I won't go into too many details, that we are using, which is very, very powerful with very high resolution. we are looking at single DNA molecules in a normal regular microscope. So you can see that each strand here is a DNA strand. And we can look at those pieces of DNA which are duplicated. I, of course, will not go into too many details. So what Asaf did, he measured the rate of the duplication of the DNA. How many letters per minute are introduced? So in the cells, before we express the human papilloma virus genes that are known to start cancer, okay? Before that, the normal cells, they do have a certain rate, 1.3 letters per minute. The number is not important. And when we express the viral genes and cells are proliferating, what we have found is that the rate is reduced. It is slower, it's abnormally slower, which this is the counterintuitive part of it. So you can see that very, very slow DNA pass we have in 35% of the strands that we measured. And it's very rare under normal conditions, 2%. Okay. So the conclusion was, as we hypothesized, that the genes, the cancer genes of the virus can lead to stress, to perturbation, to defects in the duplication of the DNA in the cell they infect. And the question was, why is that? Why there is a stress? What is wrong? What is insufficient? And we turn out to some knowledge that we had from other fields, and it's very, very trivial. It is known for many years that the duplication of the DNA is depending on those building blocks that we described, A, G, C, and T. Okay? And this pool of letters is normally very, very low, and it should raise when the cells start to proliferate. Okay, so we measured what happens in a cell that is enforced to proliferate by cancer genes, and what we found is that the level of these A, T, C, and G is going down instead of up. So they are not increased but decreased. So we hypothesized that the breaks and the mutations that we see in early stages of cancer development are because there are insufficient letters to support the duplication of the DNA. So as a geneticist, what we decided to do is to supplement the cells with extra A. Instead of T, we used U, C, and G. So we expressed the cancer genes, but in this time we supplemented the cells with extra letters. And you can see here that actually we corrected the stress. Here is the normal rate, here is the reduced rate because of the cancer gene, and you can see here a very, very high rescue. It's almost normal. So this led to much less breaks. We have a measure of DNA breaks that we talked about. So it's very low in normal cells. It's very high in cells that are expressing the viral genes for a couple of weeks. And you can see that we can recover, not fully recover, but have a major recovery. Then we asked whether the cells are losing their cancer potential. So this is a certain test that we did, and we do see that they lost their ability to proliferate. Okay? So the conclusion is that following the viral gene expression, there is a low pool of these building blocks, which is perturbing the DNA replication, leading to the genomic instability and increased tumorigenic potential. Is it specific to human papillomavirus and cervical cancer? No. Marianne Roniger in the lab, she studied another gene, another cancer gene, which is associated with lymphomas, leukemias, and sarcoma, and the name of this gene is cyclin E. And Dan Sarni, a young PhD student who is studying another gene, RAS, which is involved in lung and cancer, and lung and colon cancer, a very strong and aggressive cancer gene. And in all cases, we got the same results. So here is the conclusion that we draw from this work. When a cancer gene is expressed, cells are enforced to proliferate, but there is insufficient building blocks. This leads to perturbation in DNA replication and to breaks. And when the paper was published about 3 years ago, we found that a very distinguished scientist wrote a preview on our paper, and actually he drew a better model, a better way of describing our results, and I will end by showing this. So what Ashok thought is, he asked, does metabolite mark the proliferation of cancer cells? And he's drawing the normal process. In the normal process, usually the signal for the cells to proliferate comes from growth factors. And then in a coordinated way, all the factors, all the components that are needed for a normal proliferation are upregulated. However, in cancer, it's a cancer gene that is forcing the cell to proliferate. It's not the regular plan. So there is no coordination, not enough activation or no activation of synthesis of those building blocks, and probably many other factors which are not upregulated. They are not changing, but the cells are forced to proliferate. And by this I will end. And here is Dan, and Maayan, and Asaf, and Noah, who is doing a very nice work on the effect of nutrition on all this. But I will not have more time today. If you join me this afternoon or tomorrow, I will explain more about this part of the nutrition. And thank you very much.